Thank you. First, I'd like to credit my colleagues Mickey Parrish and Jenny Scott, who really prepared this presentation. I've ripped it off. That's the truth of the matter. I very much appreciate Wilfredo Acasio's presentation before, all the presentations that go ahead. It is incredibly difficult to design the perfect study that validates a process or challenges a product, and getting the right people involved to do that and their capabilities is just absolutely crucial. And one thing I want to say up front is that it never hurts to contact FDA in advance of this. If you anticipate that this is something that's going to have to be reviewed by a state regulatory agency, well, once you think you've got a plan, but before you've started spending a lot of money, it would be good to sit down with that state regulator. If this is something FDA is going to have to evaluate, it would be really good to sit down with the FDA and do that. I would always say that one of the ways to work with FDA is work with it institutionally. Yes, you can call up any of us that work at FDA and say, what do you think of this? And you'll get our personal opinion. And our personal opinion doesn't bind the agency to anything. So it's always a good idea to reach out to the FDA and ask FDA to get the relevant staff and experts together to hear what you have to say. And certainly we'll comment on it and offer suggestions. And if we see any weaknesses in it, or we might even say, you know, you don't really need to do this. There's an easier way to do this. So I think that it's very important that you certainly get as Wilfredo said, get the right people and organization involved. But also, I have seen time and again people do what they believe is good work with qualified labs and come to the agency, and the agency just isn't buying it. So I don't want that to happen. I don't want people to spend money. It's very expensive to do some of these studies, and we don't want anybody to waste time and effort. First, definition. I don't think there's too much confusion about validation. This is one definition of validation. In the simplest terminology, you design your manufacturing process to produce a safe, wholesome food product that has the shelf life you want, that can be marketed in the way you intend, and you really need to make sure that if you execute properly, the process you've designed will do that. I can't imagine anybody that would want to go into the food business and not want some assurance that what they plan to do, the equipment they've purchased, the way they plan to do things is going to work for them. Now, it's a separate matter whether you pull that off every day of the week, every week of the year, but you've got to know up front that if you do your part, it's going to work. Here's another definition. This one from Codex, very, very similar. Some of the overall considerations that you need to have at top of your mind is, again, as I said, effective control of the hazards. This presumes that you're going to be able to identify all of the hazards, and as we've seen from this meeting, that's always something of a changing landscape. But if you can effectively identify your hazards and come up with a way that's going to control them, that's of paramount importance. The matter of operating in control all the time depends on the assurance and the workability of your plan. Martin Cole, many of you know Martin Cole, he used to have a slide that I liked very much. It was a slide of a mountain cliff and a plateau, if you will, and imagine, if you will, that your vision was impaired. How close would you walk to the edge of that cliff? Well, obviously, if you get too close, you're going to fall off of that cliff, and you don't want food processes that operate too close to the edge. You really want to validate processes that are quite some distance from the edge, and that helps you sleep at night. If you're the person in the business that's responsible for running this company that employs lots of people who depend on you and retailers that sell your product, don't walk close to the edge, because it's inviting disaster. You want a robust process, and you want to be able to prove it. It's nice to have at your fingertips the scientific, technical dossier, the rationale of why your product works and why it can operate well within control. FDA is developing some guidance on validation that may prove helpful. We currently use the NACMCF guidance for Inoculated Pack challenge study. I was a part of writing that guidance when I was with NACMCF. Some of other people here in this room are also a part of it. I will tell you, you cannot produce a volume of any size that's going to completely cover all aspects and answer every questions and think of every contingency. God knows we gave it our best shot to produce a manageable, usable document, but as good as I think that NACMCF document is, it's no substitute for all of the things Wilfredo talked about, of having that kind of expertise and capability to do it right. Because these studies are so expensive, your first plan in validation would be to decide, can I validate this without actually having to do a bunch of studies and research and really spending serious money? And you can look at existing well-validated processes and standards and find out if you can operate according to those standards and limits. If you can, by all means do it, because those things usually have a lot of history behind them, and it's your first choice. There's a lot of good information in scientific and technical literature that can be valuable. You can use a lot of maybe pre-existing experimental data. The ICMSF books are an excellent resource. I'll say as a hint, if you're ever out there talking to a qualified individual or a process authority and they are unaware of the ICMSF books, keep moving. Find somebody else. That I personally could not get through a year without those references, and I'll bet my colleagues in here couldn't either. Mathematical modeling is becoming very effective. IRISK is quite good. ComBase is very good. One of your first stops in looking at a process before you start doing challenge studies and a great deal of work ought to be to check out what the ComBase model tells you. It can be extremely helpful, and again, if you can work with that model and engineer your process so that it operates, within those parameters of demonstrated or predicted safety, I will tell you, regulators consider that. I mean, we have a great deal of experience with ComBase, with the pathogen modeling program, with IRISK. I think we know a lot of its pitfalls, and that's certainly something you should look into. As a last resort, of course, then you may very well have to generate new scientific information and conduct a valid experiment. It never ceases to amaze me. I have seen people who want to do something that clearly hasn't been done before. They need new data. They come to some experts in the field, and the expert says, your process isn't going to work, okay? And they say, well, let's just try it. Let's design a study and do it anyway. Let me tell you what, these studies are expensive. I have seen you can design a study quite easily that could cost $100,000 to run. For God's sake, if the expert's telling you, I know it isn't going to work, it isn't going to work, it isn't going to work. Okay, most of the experts I know, they're not out to disaffect you of what you want to do. They're out there to help you, and I've seen this done. I've actually been in situations where the expert said it wasn't going to work. FDA said, we don't think it's going to work either. They do it, and guess what? It didn't work. I'm going to move through this pretty quickly. A lot of this has already been said. There's a lot of literature and experience out there. It can be hard to get for the non-expert, but whoever you're working with, really, this is part of their due diligence, is coming to you with an assessment of existing literature and existing controls and limits and requirements that you might be able to use, and what sorts of processes have been out there, and whether or not they're tried and true. FDA documents, guidance documents can be of value. The agency is more than ever in its history trying to produce good and relevant guidance documents. If you went back many decades, we didn't have very many guidance documents, and now there's quite a bit more being done. Someone mentioned the food code earlier. Well, the food code, while it's written for retail food service kind of establishment settings, it's a valuable piece of scientific thinking, because it reflects a lot of consideration about hazards and control limits, and you can also see some of these things in the things like the seafood HACCP documents that the agency has produced. It may or may not be directly applicable to your situation, but it's certainly worth looking at, and quite easy to get to. And I've already mentioned the ICMSF books. The experimental design is crucial. How many strains do you use to inoculate? What strains do you use to inoculate? How do you inoculate? How many replicates do you use? Is 5 strains better than 3 strains? Yeah, probably, but maybe 3 strains is enough. Maybe sometimes you ought to have 10. Again, some of the factors that go into that is uncertainty on the microbial diversity, or uncertainty about how one strain may behave over another. There are pitfalls aplenty in doing challenge studies and shelf life studies, and one of my mentors from long ago at Campbell Soup Company, Dr. Bud Denton, told me, he said, the cheapest thing you can do is plan. And he's right. And so I want the industry to have the best possible experience with validating processes, and good planning, lots of discussion is absolutely key to this. Recognition of all the intrinsic factors. It's just a disaster if you go through and do an experiment and you haven't looked at all of the relevant factors, the things that you need to measure: pH, water activity, oxygen tension. It can all make a difference. I had a very interesting and somewhat bitter experience in my career one time. We had lots of experience with hot fill and hold processes with jarred spaghetti sauce. Okay, the company had been producing jarred spaghetti sauce for years. We knew the temperatures. It was a well-validated, tried and true process. marketing came along. Phil's smiling because he probably knows about this. The marketing came along and said, you know, we want to start selling this stuff in flexible pouches. Great, shouldn't be hard. We find a flexible pouch machine, filling machine manufacturer, a very reputable one, and you find the film that can withstand the kind of heat that you need, and you think, well, you know, I'm going to use temperatures very similar to what I've been using in glass jars. I'm going to fill these things, this film hot. I'm going to run it through a water spray tunnel with hot water and then cool it down. I'll have exactly the same thermal parameters that I had with my jarred spaghetti sauce. Everything will be fine, right? Right. What could go wrong? So off we go. And stuff starts spoiling. And I mean it spoiling a lot. The organism that was causing it was Bacillus amyloliquefaciens. And it was doing what amyloliquefaciens does. It was liquefying the starch. It was digesting the starch in this thing and making this stuff look real runny and really not the kind of spaghetti sauce we wanted to put on the market. And you're thinking, why is this happening? Well, make a long story short, this film that we had, they got the idea that we needed to print the label on the film. And so that required that you align the film in the filler in such a way that the label would be nicely in the center of the front of the film of the package. And in order to do that, you wound up with an air bubble in the package. And that air bubble was fairly big. You know, it was probably, I don't know, 50 milliliters of air or so. It certainly didn't detract from the package, but it was a consequence of having to align that film a certain way. Well, what we found out is that if you got more than about 25 milliliters of air in that package, amyloliquefaciens, being a spore former, was not destroyed by this hot fill and hold process. And being rather aerobic, it had just enough oxygen to kick up and go and screw up your product. And this was never a problem in glass jars because of the vacuum created in the headspace of that jar. Now see, I've just given you $1 million worth of advice because that's about what it cost the company to toss all of that stuff. And they were very unhappy with the process authority in the company for this. But you know, I make my point. This is a really complicated field. I personally like, and I think the future of most validations is mathematical modeling. And the ComBase model gets better and better. And I think that what I would say to the community of scientists out there is that we all really need to work on mathematical modeling and in order to be able to do as much of this as we can without having to resort to, as I've said, costly and risky experimentations. The NACMCF challenge study protocol is important. I would use that if you're doing challenge studies, shelf life studies, I would use that as my basis. I would still meet with the regulatory agency and discuss it, but if you're not mindful of that document, you could very well find that you have an unacceptable situation. One of the problems, too, is a lot of manufacturing systems out there were not designed to be well monitored and well controlled to modern scientific standards. And that could be a real problem. And it's important to know if you have a moving cold spot in something like a dry roaster. It's important to know if variations in humidity or atmospheric temperature can be a factor. All of this has to be given quite a bit of consideration. FSMA. FSMA is the regulation we probably should have had decades ago because it really makes it clear about responsibilities for food safety and what the manufacturer needs to do in a world where we have the science and tools to understand what is going on. And again, FDA doesn't have a list of qualified individuals, but you've got to be sure you're working with qualified people, and you've got to collect your data in a scientifically valid way. Don't fear that the regulator is going to be an obstacle here. The regulator is always going to look for good science, good thinking, and everything that you can think of having been addressed, recognizing the complexity of it. It is not always possible to test to failure. It is often, though, possible to look at worst-case scenarios. If you run a test and come to the regulator with your results, whatever your operating parameters are, is the regulator is going to say, those are your limits, okay? So whatever you test, you need to design it to be a worst-case scenario or a test to failure so that you know how close to those limits you can safely operate. If you run a test and it's quite dependent on pH, and your maximum pH is 4.2, it will not be acceptable to a regulator for you to run at 4.3. So you need to know where you need to be, and you need to set these limits up within reason. It's possible to design such a worst-case scenario that your whole experiment fails. You've got to use very good judgment, have a operating window, yet be able to, once you're done, have some comfort that you can run each day without exceeding your limits. A lot of this has been talked about by others. Replication is important. It's very difficult to just do a thing once. And all of us in science laugh at the magic number 3, you know. I would say sometimes 3 replicates is not enough. It's very, very rare that fewer than 3 replicates is enough. You have to take large enough sample sizes to be able to detect small numbers of surviving microorganisms, if that's the nature of your study. And you have to be sure that your methodology works with your matrix. It's very important, if you're going to do this study, document well. I have seen a number of cases where a study was probably competently done, it was a costly study, and the laboratory that did the study just did a lousy write-up. It was so brief that you really couldn't tell what they'd done. There was no raw data in there. You'd look at it and just, you know, and I think that occurred because the lab was used to doing different kinds of work and writing brief, to-the-point reports. But, you know, you can't come to FDA with a one-pager that says, we did this challenge study and all the results were acceptable. We want to see all the details. We're going to probably want to see the raw data. We're going to want to understand the methods. We're going to want to see what your controls were. So if you're a lab or a consultant doing this kind of work for someone, factor in the time you're going to have to spend writing what might amount to a mini thesis. Needs to be done. I think that the study, in summarizing, the study should really be done with the view to it's going to be put into operation and the records are going to be kept, and what really should flow from the study is what operational limits will be used in actual manufacturing and what records will be collected in actual manufacturing. I think that would be my final thought, is this all has to be done with the manufacturing feasibility in mind. I'm sure you have some questions for me. So, the vision for FSMA implementation is that FDA will be adequately staffed with subject matter experts that you can come to who can address your concerns and properly evaluate this. I can't tell you that there is a currently organized portal of entry into CFSAN, okay? I think what you have to do is the vast majority of this is probably going to fall in CFSAN's Office of Food Safety. And so I think probably that's the first portal of entry to FDA would be to contact its Office of Food Safety. You can also, I mean, you can never go wrong contacting the local district, and then the local district will engage CFSAN and they'll make sure that the proper people are there. But I think most of it, you know, go to the Office of Food Safety.
So Larry Keener, International Product Safety Consulting. Thank you, Don, for an excellent overview of the regulatory view to validation and the regulatory expectations for validation. As you know, there are a number of us working on novel manufacturing, pasteurization, and sterilization technologies. And I was wondering if you could talk a little bit about the agency's expectations in the master file document to support, for instance, pressure-enhanced sterilization or post-electric fields or superparamagnetic nanoparticles for food sterilization.
When you come up with a process that's completely novel, and we've seen these over the years, things like high pressure, that's a situation where trying to devise a test to failure is quite useful. I think that you don't necessarily need test to failure for everything you might do, but boy, when you have a novel process, knowing where it's going to fail, where it's going to break down, I think is one thing that I am particularly interested in. You can compensate for that to some extent by having processes that are, you know, where you're challenging it with such a large number of microorganisms that perhaps that's buying you some margin, but test to failure is pretty hard to substitute for.